Today we will be talking about transcription in prokaryotes. The enzyme RNA polymerase copies the DNA strand by making phosphodiester bonds in 5' to the 3' direction to join nucleotides together. RNA polymerase does not require a primer to begin synthesis. The steps are as follows. To explain the next steps of transcription, I will have the DNA strands uncoiled and have the coding strand on top and the template strand on the bottom. I am adding three aspects for this diagram: the -35 site, the -10 box, and the +1 site. Together they form the promoter region. The +1 site is also the initiation site of RNA transcription, and the RNA coding region is the +1 site combined with the termination spot, which is the termination of transcription. The -35 site and the -10 box are also a consonance sequence. The -10 box is also called a Tata box, and both will have similar gene sequences in most prokaryotes. Their function is to recruit RNA polymerase. The core enzyme and the sigma factor are added to the diagram during the process of template binding and chain initiation. These two factors attach to the promoter region, and combined they are called the helo enzyme. Once bound to the RNA polymerase, helo enzyme separates the DNA strands, creating a transcription bubble. The RNA polymerase has found the promoter region and has started transcription. Then the sigma factor leaves. When the sigma factor leaves, elongation begins. RNA polymerase needs a template strand to build an RNA strand. The template strand reads 3' to 5', while the coding strand reads 5' to 3'. The coding strand has the same nucleotide sequence as the RNA strand, except uracil takes the place of thymine. These bases are added to the RNA by cleaning two phosphate groups from R-NTP from the phosphodiester bond. The last step in transcription is termination. It occurs in two different ways. The first one I will talk about is rho-dependent. Rho-dependent termination occurs when the Rho factor protein binds to the RNA sequence and moves towards the 3' end of the RNA. Then a helicase enzyme is used to unwind the DNA in an RNA connection, causing termination. In rho-independent termination, inverted repeats are present in the DNA, causing the RNA to form a hairpin structure. The hairpin inhibits RNA polymerase, causing it to stop. This causes uracil to form in RNA. The weak bond between adenine and uracil causes termination.